I am going to present to you Claudia Di Berardino, who is a student of the doctoral program of Neuroscience from the University of Valparaíso. She born in Atri, a beautiful place from what I found in internet. She follows her career at the Alma Mater Studiorum University of Bologna, the oldest university of the world, which is very cool if you ask me. She continues her master's degree studies in Milan at Vita Salute San Rafael University. She is interested right now in the modulation of the retina, specifically by serotonin. And she can share with you more information about that in the talk, Serotonin Modulation of Spontaneous Activity, Transmission, and like evoked response in the mouse inner retina. Then, to Claudia.
Okay, thank you, Scarlett, for your nice introduction. So I'm going to share the screen. Okay. Please let me know if you can see the presentation.
It's black right now, but it's sharing.
Okay.
There it is.
It's a little bit slow. Here. Okay, perfect. So, good morning, everybody, and thank you all for being here today for my presentation. And today I'm going to show you some of the work I've been realizing in the, oh, maybe I should do that. Okay. In the laboratory of Professor Andres Chavez. And as some of you may already know, my work is aimed at unraveling the role of serotonin as a neuromodulator in the retinal circuit, with particular attention to the regulation of the function of retinal ganglion cells, which are located in the inner retina and that convey the retinal output to the brain targets, to the brain centers. And so I will start with a brief introduction of the retinal circuit, and then I will show you some evidence of the serotonergic system playing a role in this circuit. So as you know, the retina is the first station of, I'm sorry, can you see the title? Because
yeah, I can see it.
Okay, perfect, sorry. So the retina is the first station for detection and processing of visual stimuli from the external world. And this thin tissue here in blue, located in the back region of the eye, where it receives light stimuli through the eye and converts them into meaningful information for the organism, codifying the different aspects of the visual scene, such as shape, color, and movement. And this function is mediated by the the coordinated work of multiple neurons that are precisely organized in the different retinal layers that has these typical laminar structures based on their specific function. For example, photoreceptors are located here in the outer layer of the retina, where they transduce light stimuli into neural signals that they transmit to bipolar cells here in the inner nuclear layer that then makes synaptic contacts, excitatory contacts onto retinal ganglion cells here in the inner retina, whose axons form the optic nerve that sends the retinal output to the brain visual centers. And importantly, this vertical excitatory pathway is finally regulated by two types of interneurons, of inhibitory neurons, that are horizontal cells and amacrine cells here in the inner retina. And it is in this region, in the inner part of the retina, where the integration of synaptic inputs, both excitatory and inhibitory, occurs at the level of retinal ganglion cells, or RGC, as I will call them from now on. And this process is regulated by different neuromodulators, such as dopamine, endocannabinoids, nitric oxide, and also serotonin, whose role, however, in this tissue is very poorly understood today. And this is why we focused our study to further understand it. Notably, all the components of the serotonergic system are expressed in the vertebral retina, starting from the enzyme for its synthesis, the tryptophan hydroxylase, the vesicular transporter BMA2 that is responsible for its storage in exocytotic vesicles, the membrane transporter SERT that mediates its reuptake from the extracellular space, and also several subtypes of serotonergic receptors with a high level of species specificity and also a difference in their expression levels during the development. And in this summarizing cartoon, I want to focus on 2 points. So the expression of SERT that is distributed along in the inner retina at the level of ganglion cells, and also it is expressed by a subclass of bipolar cells and endocrine cells that have been reported to be able to accumulate serotonin here in the retina. And also I want you to focus on the fact that the synthetic enzyme is expressed only by photoreceptors here in the other retina, where it is used. Serotonin is used as a precursor for melatonin production, and only recently it has been described that TPH activity is also present in a subclass of amacrine cells, so demonstrating that this group of cells can synthesize locally serotonin, and so could be the local source for serotonin in the retina. Moreover, it has been reported both in rodents and in primates that there is a reciprocal connection between the raphe and the retina, where the serotonergic projection from the raphe nuclei innervate the inner part of the retina, and particularly the ganglion cell layer, as we can appreciate in this 3D reconstruction. And here, these terminals could be releasing serotonin too. So we have 2 potential sources of serotonin in the retina, a local source from amacrine cells, and also an external source from the terminals of serotonergic raphe projections. And so even if all of the components of the serotonergic system are present here in this tissue, the role of serotonin as a modulator of retinal synapses has not been studied, and there's very few evidence for a functional role of serotonin modulating retinal ganglion cell function. For example, there's an early study in the rabbit retina where it was shown that 5-HT1A receptor agonists and 5-HT2 receptor antagonists can induce a reduction of the spontaneous firing activity of retinal ganglion cells, and also the on component of the light evoked response in these cells. And so this study suggested that these 2 classes of receptors, the 1A and the 2, could play an opposing role in the regulation of RGC function. Moreover, they showed that these 2 pharmacological agents were not able to affect the properties of horizontal cells, nor the light evoked response of bipolar cells, thus meaning that they could act directly in the inner retina. Okay? And they could be regulating directly the RGC excitability or the synaptic inputs they receive. And there's a recent study supporting this last mechanism, providing evidence for this mechanism. In fact, the group of Zhu et al. Showed that the activation of 5-HT1A receptors is able to increase the GABAergic input to retinal ganglion cells. However, if this receptor contributes also to the regulation of excitatory transmission or of the light evoked responses in retinal ganglion cells was not investigated. And also it remains unknown how the endogenous serotonin is able to regulate retinal synapses. And that's why we aimed our study at answering these questions and elucidate how serotonin modulates retinal ganglion cell function, with a particular focus on the role of endogenous serotonin and its regulation by SERT, together with investigating the receptors involved. And so, to this purpose, we use both wild-type mice and a known genetic model of serotonin homeostasis disruption, the SERT knockout model, to evaluate if altered levels of serotonin in the circuit could affect retinal physiology. And so, to answer this question, We used the third knockout model that was kindly provided by Professor Pablo Moyas here at CMB Institute. And importantly, in this model, the expression of the SERT transporter is lost due to targeting gene disruption here. And so, if you remember, SERT is responsible for reuptaking the serotonin molecules from the extracellular space. So it finally regulates serotonin concentration here at synapses. So if this homeostatic function is lost, not only serotonin levels are altered, but also there is a global dysregulation of all the function mediated by the serotonergic system. And in fact, this model, there are not only changes in the synthetic and degradation rate of serotonin and a global and transversal change in the expression and activity of all the receptors subtypes. And these changes have been well characterized over the last 2 decades to study the basis of the anxious and depressive behavior that this model shows. But what happens, even if it has been studied a lot in many brain regions, what happens in the retina of this mouse has not been investigated. And I hope that by now I've convinced you that the serotonergic system is expressed in the retina. And so we went to investigate what happens in this circuit when serotonin levels are altered. And so we use wild-type SERT heterozygous and knockout mice of 1 to 2 months of age and combine biochemical, histological, and electrophysiological techniques to evaluate if the retinal function was affected in these mice. Here we can see that our collaborators validated the model, demonstrating that the serotonin global content in the retina was significantly reduced, as was the content of its metabolite 5-hydroxyindolacetic acid. In the heterozygous, we can see that the level is intermediate, thus reflecting a gene dosage effect. But even if the serotonergic levels were altered, we didn't appreciate any changes in the gross morphology of the retina. As we can see here, there was no significant difference in the layer thickness and also no differences in the neuronal density in the 3 nuclear layers. But even if the macroscopic structure was not altered, when we analyze the global circuit activity by recording RGC firing in the whole mount retina by MIA recordings, microelectrode recordings, we observed a significant reduction in the global firing rate, as we can see here, both in scotopic and photopic conditions, and also following white noise stimulation, suggesting that the circuit function is altered by as a consequence of the altered serotonin levels. And then we asked if this could be associated to a reduced neuronal excitability or to reduction in the synaptic transmission onto retinal ganglion cells. And to answer this question, we performed host cell patch clamp recordings of individual retinal ganglion cells in acute retinal slices, transversal slices, as we can see here, both in the current clamp and voltage clamp mode, to evaluate RGC excitability and the synaptic inputs onto retinal ganglion cells, respectively. So we first analyzed RGC excitability, and as we can see here, we didn't observe any changes in the resting membrane potential of these cells, nor in the voltage threshold for firing, nor in the heterozygous, nor in the knockout retinal ganglion cells when compared to wild type. And moreover, when we analyze the voltage responses to a current step protocol, as we can see here, we also couldn't observe any differences in the responses to hyperpolarizing and depolarizing steps, nor in the firing frequencies of these cells, nor in the action potential adaptation. So altogether, these results demonstrated there are no significant changes in retinal ganglion cells excitability due to the alteration of extracellular serotonin levels. So we asked if the synaptic transmission was altered, And so we recorded spontaneous excitatory and inhibitory postsynaptic currents in retinal ganglion cells. And as we can see here, the excitatory input was not altered, both in the heterozygous and knockout mice. In fact, we couldn't observe any changes in the frequency nor in the amplitude of the excitatory events. And when we analyzed the inhibitory input, we couldn't observe any changes either. So no changes in the mean frequency nor in the amplitude. So suggesting that there are no changes in the synaptic inputs that retinal ganglion cells are receiving, but the changes that we observe in the MEA are can be appreciated only at a circuit level. So to summarize this first part of results, till now I showed you that serotonin homeostasis is impaired in the retina of certain heterozygous and knockout mice, as shown by the significant reduction of retinal levels of serotonin. And this confirmed that SERT has an essential function in maintaining retinal serotonin levels in a physiological range, as it does in other neural circuits. Moreover, I showed you that this alteration in serotonergic levels is associated with a reduction in RGC global activity, suggesting that the outputs sent to the brain area might be altered, and this would be very interesting to further investigate. And this also indicates an important role of endogenous serotonin in the regulation of retinal physiology. However, when we analyze the excitability and synaptic inputs of individual retinal ganglion cells, we observed that they were not altered in certain heterozygous and knockout mice when compared to wild type, indicating that the defect in RGC activity seen in the mirror cortex can be appreciated only in an intact retinal circuit. So, till now, I showed you that maintaining serotonin homeostasis is important for normal retinal ganglion cell function, but we still don't know how the serotonergic system regulates retinal synapses in the healthy retina, because till now we were using a constitutive knockout model with all the limitations that are associated to it. So in parallel, we also wanted to investigate how serotonin regulates synaptic inputs onto retinal ganglion cells in wild-type mice. And I'm showing now to you some results about the regulation of excitatory synapses only. So, first of all, we wanted to evaluate if increasing the levels of extracellular serotonin in the extracellular space here by using pharmacological agents that target SERT, for example, like fenfluramine, we could produce some changes in the synaptic transmission in the inner retina. And so, first of all, we use fenfluramine, that we know that is an agent that induces a massive release of serotonin from the intracellular, from the terminal, by inverting the flux of serotonin through SERT. So instead of reuptaking it, now it is releasing it. And after application of fenfluramine to the bath, we could observe a marked and significant reduction in the frequency of spontaneous excitatory currents without any changes in their amplitude. Then we also use citalopram, that is another drug that targets SERT, but with a different mechanism. As you may know, the citalopram is a selective serotonin reuptake inhibitor, and so it blocks the reuptake through SERT. Okay, so we also will have a higher concentration of serotonin in the extracellular space, but not as massive as with fenfluramine. And as we can see here in these preliminary results, we also observed a reduction in the frequency of both spontaneous excitatory currents and minis without any changes in the amplitude. So altogether, these results showed that increasing the extracellular levels of serotonin could reduce the frequency of spontaneous excitatory postsynaptic currents, and that indicates or suggests that endogenous serotonin could be modulating excitatory synapses through a presynaptic mechanism. And so given this interesting observation, we next asked if endogenous serotonin could also be regulating light-evoked responses in retinal ganglion cells. And so we recorded evoked postsynaptic currents in retinal ganglion cells after or during light stimulation. And we analyzed the evoked response, recorded both at plus 40 millivolts and minus 60 millivolts. This was to evaluate any potential differential modulation of the AMPA and NMDA receptor modulated currents by serotonin. And before showing you the results, I wanted you to understand how we grouped the retinal ganglion cells based on the light evoked responses. So here we can see the retinal ganglion cells can receive synaptic input, excitatory synaptic input from on bipolar cells here in the inner part of the retina of the IPL, or from off bipolar cells in the outer part. In this case, this would be an on-off cells because they receive both contacts. Here, I'm showing you not only the morphology of these cells, but also how you should see the light evoked response. So in a non-cell that received an excitatory input from on bipolar cells, they will show an excitatory response at light onset, as we can see here. So in current clamp, we should see firing here a light onset and an excitatory postsynaptic currents in voltage clamp mode. And off cells, on the other side, we show an excitatory response at light offset. So with a firing at the left offset and a current at the end of the light stimulation. While on-off cells will respond at both light onset and offset. So now we show you the effect of endogenous serotonin of these excitatory postsynaptic currents that look like this. So after back application of fenfluramine, what we could appreciate was a significant reduction, a marked reduction of the amplitude and also the synaptic charge of the EPSC evoked, both in on cells and also off cells. And importantly, we could record the response in off cells, both at plus 40 and minus 60 milliwatt, where we observed in both cases a drastic reduction. That's showing that supporting the idea that both NMDA receptor mediated currents and AMPA mediated currents are affected, and strongly supporting the fact that the modulation is presynaptic. Moreover, I wanted to show you that after 30 minutes washout, the amplitude and also the shape of the response was completely recovered in this example that I show you here. So, summarizing this second part, I show you that increased extracellular levels of endogenous serotonin can induce a reduction in the excitatory spontaneous synaptic transmission onto retinal ganglion cells, likely through a presynaptic mechanism. That increased level of endogenous serotonin can also reduce the amplitude and synaptic charge of the light evoked EPSC in retinal ganglion cells, and that those results indicate that by regulating the extracellular availability of serotonin, SERT is playing a major role in regulating synaptic efficacy in the retina. So I also told you that I would have shown you some results about the receptors contributing to this effect. And now I'll show you some results about the involvement of the 5-HT1A receptor that was our first candidate, not only because its expression was reported in the inner retina, and also because there were functional evidence of its ability in the rat to modulate synaptic transmission, but also because in other neural circuits, it has a known inhibitory role. And so we applied this selective 5-HT1A receptor agonist, that is H-hydroxydipat, and recorded its effect onto the spontaneous excitatory activity. What we observed was a marked reduction of the frequency of a CPSC without any changes in their amplitude. This was true also for MINI, further supporting a presynaptic modulation. So the effect of H-hydroxydipat is mimicking the effect induced by fenfluramine and citalopram. However, when we apply fenfluramine in the presence of cyanopindolol, that is a 5-HT1A1B receptor antagonist, we could still observe this effect onto the frequency, this reduction. So this was an unexpected result, and we started to think that maybe it wasn't the 5-HT1A receptor mediating the effect of fenfluramine. But to confirm or exclude the participation of this receptor, now we have posed a more specific antagonist for the 5-HT1A receptor, and now it is available in the laboratories, and we're going to test it to confirm this result. Moreover, I want to show you that the activation of 5-HT1A receptor was also able to reduce the amplitude and synaptic charge of light evoked responses in retinal ganglion cells, both in on and off cells. However, in the off type, there wasn't a reduction in the amplitude, but there was a change in the shape with a slight delay in the onset of the response. And this was true also when we recorded light evoked responses at -60 millivolt. So again, H-hydroxydipad is mimicking the effect of fenfluramine onto the light evoked responses. It affects both AMPA and NMDA receptor mediated currents, so probably it's acting presynaptically. But again, when we applied H-hydroxydipad in the presence of xenopindolol, we continue to observe the effect. So, to summarize this last part of the results, what I show you till now is that the 5-HT1A receptor agonist H-hydroxydipad can mimic the effect of fenfluramine and citalopram onto spontaneous excitatory events, producing a reduction in the synaptic efficacy of the excitatory input onto retinal ganglion cells, and this process is likely to a presynaptic mechanism. H-hydroxydipad also reduces the amplitude and synaptic charge of light evoked EPSC in retinal ganglion cells. However, we were not able to block this effect through the 5-HT1A1B receptor antagonist xenopindolol. So we urgently need to use a more specific antagonist in order to confirm and exclude the involvement of this class of receptors. So I would like to conclude the main results that I showed you today are that in the search knockout mouse model, altered serotonin retinal levels are associated with altered retinal ganglion cell functional output. Second, that in the wild-type retina, increased serotonin extracellular levels depress excitatory spontaneous synaptic transmission onto our disease, likely through a presynaptic mechanism, and also it could also reduce the light evoked responses in on-off and also on-off retinal ganglion cells. However, the receptors involved remain to be clarified. But to our knowledge, this is the first evidence of regulation of both spontaneous synaptic transmission and light evoked responses by endogenous serotonin in retinal ganglion cells. And these results could have implications not only in mice, like for example, in certain knockout mice, the retinal output sent to the brain is altered because the retinal global circuit activity is altered. And this could have potential consequences not only for the vision of these mice, but also for non-visual functions, given the non-projection from the retina to the brain areas that regulate functions such as mood and circadian clock. And in fact, there's evidence that altering this connection from the retina to the RFA could affect mice behavior and produce a depressive behavior. Moreover, it is important that there could be possible implications for humans, for example, for depressive and anxious patients, in which it is known that serotonin levels in neural circuits are altered, and so also retinal visual processing could be altered in these patients, with potential impact not only on their visual system, but as I told you before, also on non-visual function. And finally, there's an important question that has not been assessed, and it is if the chronic treatment with antidepressive drugs like citalopram that aimed at increasing serotonin levels by blocking SERT could affect the retinal function in patients, and this could produce important and severe side effects in these patients. So I would like to thank, first of all, my mentor, Andres Chavez, all my lab colleagues, Sebastián, Alejandro, Rodrigo, y Carlos, all our collaborators, and all the members of my evaluatory committee that have been always very supportive during the realization of my PhD thesis, and obviously all of you for your attention. And now I'll be glad to answer your questions. Someone?
Dr.
Did you hear me? Right?
Yeah. There are two hands raised. Pablo Moya was first, if I am correct.
Hello, Claudia. Wonderful
Hi,
presentation. Congratulations.
Thank you.
I just wanted to ask, have you checked a potential upregulation in any of the organic cation transporters in the serotonin transporter knockout mice, such as the OCT3 that also is involved in serotonin uptake, in case you can have a compensatory effect?
Sorry, Pablo, I can't understand you. I can listen to you. Could you please?
Sure. I'm wondering if you have checked a potential compensatory upregulation at any of the organic cation transporters that could also transport.
Yeah, you're right. No, we brought the antibody. Yeah, you're right. We know that this compensation happens in other areas. And in fact, we plan to investigate these aspects, but we bought the antibody and we still didn't check this potential compensatory mechanism, because it is known that there could be an overexpression of OCT3, for example, in the retina transporter to compensate for this reuptake of the serotonin, or that is in excess in the extracellular space. Yeah, but we haven't checked it yet.
Okay, next, I don't know if we go with Adrián Palacios or Romualdo Torre.
Okay. Very nice, Claudia.
Thank
you. I have more like a reflection of. I think about the result that you show. Comparing like single cell recording and the multi-electrode recording. And I think what need to be think about is that you have in the retina like 20 class of ganglion cells.
Yeah.
So this is the first issue. And then what could be complementary, what you have done is just to reanalyze the data from the MIA recording in terms of the clustering to try to understand which kind of the cells in the recording we are recording from, and then compare with the wild type and the transgenic mouse, which one are more present and the other. And for me, it's not a problem that sometimes you have, good recording from the ganglion cell in the single cell level, because what the multi-electrode is taking from the circuit is more like the integrity of the circuit, in the sense that you have for receptor, bipolar cells, two different classes of bipolar cell, and then you have the ganglion cell. So this is more related what you have done with light, the sense that you are recording from light, and then you take from the ganglion cell level the integrity of the circuit, that they are the ones that I think are more alterated than directly the ganglion cells in itself. So I think this is something that we should like complement, in the future, make an analysis more close to the single cell types, that is something that we know how to do now, and this is very recent. So you don't have this data today, but we can provide you with more, I mean, and fine detection of the circuits, you know, I mean, a specific circuit that should be more, I think more deteriorated than the other, if is that the case.
Yeah, it would be wonderful because I also tried to separate my recordings into on-off and on-off cells, but I couldn't see any differences. So it will be very important to understand if there is only a subclass of cell that is affected, because as you tell us, there are more than 30 retinal ganglion cell types. So it could be possible that in my recordings, this defect is masked because I pulled all the cells together.
Yeah, very nice. Thank you very much for your talk.
Thank you.
Then, Professor Doctorado, do you have any question? Pamela, ¿tienes tu pregunta?
Sí, hola, buenas tardes.
Hola.
A ver. Ya, disculpa, voy a preguntar en español. Claudia,
Sí,
¿podría
sí,
devolver a la diapositiva, la última, por favor? Quería preguntar de ahí una cosita.
¿De esta o de la última de los datos?
No, esa, la última.
Ok.
Sí. Finalmente me quedé con una duda. Quizás mi defecto en inglés, que puede ser que no le he entendido bien. Pero en el tratamiento crónico con antidepresivos, entonces, finalmente, ¿cómo afectaría, según este estudio, la función de la retina? No me quedó claro.
Mira, en realidad eso ha sido investigado muy poco, en el sentido que existen solamente estos dos reportes de casos individuales. En un caso, aquí es un hombre de 23 años y aquí una mujer, y lo que se ha visto es asociado al tratamiento con sertralina una maculopatía bilateral, que significa que se empezó a desarrollar después de, en un caso después de un mes y en el otro caso después de 6 meses de tratamiento, una lesión de la mácula, ¿ya? Y eso significó también una pérdida de la, ¿cómo se dice? Visual acuity, de la...
¿Agudez visual?
Agudeza visual.
Sí. Gracias. Y entonces se vio que en un caso, después de parar el tratamiento, en el caso del hombre que había tenido un tratamiento más corto, se pudo recuperar la agudez visual, mientras que en el caso de la mujer que había sido tratada por 6 meses, la lesión desapareció, pero el defecto en la visión permaneció, no mejoró. Entonces, esos son los únicos reportes, pero según yo, valdría absolutamente la pena de ampliar este estudio, porque normalmente, obviamente, las terapias y también los médicos se enfocan en evaluar los síntomas psiquiátricos, pero estamos viendo que también los cambios en el procesamiento de las señales en la retina puede enviar señales alteradas también a otras áreas que regulan también el ánimo, el estado de ánimo y todas las funciones relacionadas. Entonces podría ser una comunicación recíproca que se está alterando y bueno, también aumentar los niveles de serotonina aquí a nivel de la retina podría ser no tan bueno. No sé si respondí tu pregunta.
Sí, súper. Y relacionado con lo mismo, entonces, en depresión y ansiedad se altera entonces el sistema visual, ¿verdad? La segunda parte. ¿O no? ¿Se sabe? No, esa parte no lo entendí bien.
Se debería estudiar, porque en realidad en humanos, en pacientes, se ha investigado súper poco. O sea, no hay una relación directa, la verdad, no existe. Y también en los modelos animales, generalmente en los estudios uno se enfoca más en la conducta y sobre todo en los síntomas psiquiátricos, o psychiatric-like, porque son animales. Entonces no es claro lo que estaría pasando a nivel del sistema visual, sobre todo a nivel de la retina, ¿ya? Porque después pueden haber también, no sé, un cambio compensatorio a nivel de la corteza visual que podrían compensar esta alteración en la retina, pero no lo sabemos.
Muchas gracias.
Lo siento.
No, yo disculpo porque no lo hice en inglés. Gracias.
No, tranquila.
And then we go with the question from Professor Latorre, who muted himself a minute ago.
Claudia, very nice talk.
Thank you.
I am up to the ignorant in this field, so my question is just curiosity. Did I understand correctly that one of the compounds make the serotonin transporter to work in reverse?
Yeah, fenfluramine, yeah.
Huh? Yeah, so and you know, everything is know about the mechanism about that. What that happened, because the transporter is essentially passive transport, no?
Yeah, sure. What happens is that fenfluramine is a substrate analog. So it has a very similar structure to a serotonin. So fenfluramine enters through SERT, so it takes advantage of the transporter, and it induces the inversion of the flux. So this is because when you increase the fenfluramine levels inside, so serotonin also increases in the cytoplasm, and so the gradient of serotonin is increased in the cytoplasm, and also it is dependent, this flux from on a current that is associated, an ionic current that is associated with this flux. But primarily it is because fenfluramine accumulates inside the cells, and so serotonin is exiting.
Okay, thank you. Very nice.
Mm-hmm.
Great answer. Thank you.
And is there another question for Claudia? Well, if that's not the case, then we will close this session, and we want to thank to Claudia for the nice talk and to your presence here. Thank you.